Today we're talking about how probability applies to genetics so we can predict the likelihood that particular offspring will occur in the future. Our success criteria for today is being able to predict the probability of simple Mendelian traits using a Punnett square. So what is a Punnett square? Punnett square is our way of visualizing probability in genetics. What it is it's a square with four quadrants when we're looking at a single trait, and each of the quadrants represents a possible offspring. Along the top and along the side of the Punnett square, we are going to write the genotypes of the parents. Now you'll notice that we only put one allele in each column or row. We don't write big R, big R, little r in this example at the top of each. We only write the big R or the little r. And the reason we do that is this actually represents this independent assortment of the alleles and how there's only one possible allele that can be passed on in a gamete. So then, using the Punnett square, we can visualize what the different possible offspring are by saying, okay, what if I take this R and combine them together? So if you get little r from both parents, you get a homozygous recessive. Or you can get a big R and a little r two different ways. And you'll notice that even though the order that you get the big R and the little r is different, whether or not it comes from mom or dad, we still write it with the big R first to make sure we're indicating that these individuals are heterozygotes and they actually have the same uh, genetic makeup there. And of course, if you get a big R from both parents, you get a homozygous dominant. So, the reason we want to use Punnett squares is because genetics is probability. The likelihood of a given allele being passed on in a gamete is a random event. Since there's two possibilities, that means you have a 50-50 chance of either allele getting passed on. So a very common metaphor we use for genetic inheritance is flipping coins, because fair coin works the same way. You have a 50-50 chance of getting head or tails. So if we know the parents' genotypes, we can predict the likelihood of specific genotypes and specific phenotypes occurring in the children. We can express those likelihoods in four different ways: as a probability, which is a decimal between 0 and 1, as a percentage, which is, as we know, between 0 and 100. We can represent it as a ratio as well, or we can represent it as a fraction. So, what are the rules that we need to use when we're talking about inheritance? The big rule is the rule of multiplication. The probability of independent events occurring simultaneously is the product of their individual probabilities. So we can multiply the probabilities of two events happening together to find out what the likelihood of both events happening is. Important thing when you're using the rule of multiplication, you either need to be using a probability between 0 and 1, or you need to be using a fraction. If you try and do this with a percentage or a ratio, it's not going to work. So here's an example question. What is the probability of a homozygous recessive offspring occurring when two pea plants that are heterozygous, big P, little p, for flower color breed? If you want to pause the video here and work it out on your own, go for it. I'm going to continue in 3, 2, 1. So the way that we're going to figure this out is we need to figure out the probability of the sperm in the egg receiving a particular allele. And this exact example is actually represented in the diagram I have at the side. The probability of the egg receiving small p, the recessive allele, is 0.5, or 1 out of 2. The probability of the sperm receiving a small p allele is also 0.5, or 1 out of 2. So we can multiply those two probabilities together. And that will tell us what the probability of both the egg and sperm fertilizing with those alleles will be. So 0.5 times 0.5 gives us a probability of 0.25. Or 1/2 times 1/2 gives us a probability of 1/4. Once we have those numbers, we could also times the probability by 100 to give us a percentage. So you want to pay close attention on a test if the question is asking for probability or percentage, because those will be two different answers. So we can multiply by 100 to turn a probability of 0.25 into a percentage of 25%. We can also get a ratio out of the fraction. Now the ratio here is going to be a 1:3 ratio. There's going to be one homozygous recessive individual for every 3 non-recessive individuals. So we actually write it as 1/3, not 1/4, because 1/4 would actually mean there's 5 possible outcomes, and our denominator in our fraction is 4, so in the ratio everything has to add up to 4. We can also use Punnett squares to figure out what an unknown dominant genotype is. We call this a test cross. If we have an individual that we observe the dominant phenotype in, we have no way they're a heterozygote where the dominant allele is overpowering the recessive allele. But if we can breed them with an individual that we know is homozygous recessive, that will actually tell us if they are homozygous or heterozygous for the dominant trait. The reason that this happens is, again, probability. If the unknown individual is homozygous dominant, the only allele they can pass on is the dominant allele. So all of their offspring will show the dominant phenotype. However, if they're heterozygous, half the time they'll pass on the dominant allele, and half the time they'll pass on the recessive allele. So, instead of all of them showing the dominant phenotype, if they're heterozygote, half of their offspring will have the dominant phenotype and half will have the recessive phenotype if you breed them with a homozygous recessive individual. Now, obviously, if you only do this for 2 offspring, there is a chance that you will get both dominants. It is possible. So test crosses work best when you can get lots and lots of offspring, because all it takes is one recessive offspring to show the heterozygous nature. But there is always a chance, so the more tests you can do, the better your odds are of having an accurate result.